Hi everyone, and welcome to the 9th TIA Centre seminar of the academic year. So my name is Adam Shepherd, and I'm a research fellow here at the TIA Centre. And for those of you that are new to our seminars, we try and involve researchers from across the world to give give talks on new and exciting pieces of research. So we're delighted today to be joined by Georg Wölflein. So George is a PhD student in computer science at the University of St Andrews, under the supervision of Dr. Arangelovic. Sorry if I didn't pronounce that correctly. Prior to this, he completed a direct integrated master's degree in computer science at the same institution. His research lies at the intersection of computer vision, deep learning, and computational pathology. In particular, he is interested in weakly supervised biomarker prediction in histopathology images, as well as self-supervised learning and vision language models. The title of George's talk today is "A Good Feature Extractor is All You Need for Weakly Supervised Learning in Histopathology." So thanks again, George, for joining us. And yeah, with that, please feel free to start your presentation.
Awesome. Thank you very much, Adam, for the introduction and the opportunity to present at the TIA seminar series. Yeah, as you said, I'm a PhD student at the University of St Andrews. And today I'll present about some work that I carried out mainly during a summer internship at Professor Jakob Kator's lab at EKFZ in Dresden, Germany. All right, I will start this talk off by making two claims about weakly supervised learning, so specifically weakly supervised whole slide image classification, which I will try and convince you of in the remainder of this talk. And if there's anything that you take away from this talk, these are the two main points. So firstly, that stay normalization can be omitted in weakly supervised whole slide image classification, and similarly also image augmentations. I will specify what I mean by that in a second. And secondly, that ctranspath and lunetDino, which are two feature extractors, those are the two best publicly available feature extractors that we currently have. The emphasis here is on the publicly available bit. We just 2 weeks ago, we had a presentation from Dr. Gabriele Campanella from Mount Sinai, who presented their foundation model, which is also a feature extractor. But those types of large models right now usually don't release their weights. So I'm only making claims about the publicly available ones. All right, so let's start by defining what is weakly supervised whole slide image classification. A whole slide image is an image of a tissue section, which is usually around like a square centimeter in size, but once you scan it under a microscope, that size increases in terms of the number of pixels to billions of pixels. So it can be around 100,000 by 100,000 pixels. And oftentimes we have a whole slide image and some sort of label associated with that image. So we might have a patient and some information about that patient. And for a particular patient, we might have one or a couple of whole slide images. And the problem is we can't just feed this one whole slide image into a neural network in order to just classify it. We have to first split it into patches. So the first step is always splitting this whole slide image into some non-overlapping patches, performing some deep learning magic, and then you get some classification, for example, a binary or a multi-class classification for predicting a certain target. The reason this is called weakly supervised is because, of course, we have this collection of patches, but we don't have a label for every patch. We just have a label for the whole slide, so for this whole collection of patches. Yeah, so why is this an important problem? Often we have patient-level labels, as I mentioned. So we have a label for one whole slide image, but not for basically every pixel. We don't have segmentation masks or patch-level information. And this can be useful for predicting biomarkers like genetic alterations or predicting survival, all serving for the goal of increasing like personalized medicine and personalized treatments. All right, let's look at that in a bit more technical detail. How does whole slide image classification work? There's like two main phases. There's the pre-processing stage and the training stage. In the pre-processing stage, we start with our whole slide image and we split it into patches, as I mentioned. And what a lot of people, and which is currently the most widespread approach, is to first perform stain normalization. So all of the patches or the entire slide, depending on how it's implemented, you undergoes this sort of stain transformation. The reason for that is that oftentimes we get slides from different centers, different countries, different scanners, slightly different dyes, and different microscopes. So these patches or slides will vary in appearance. And in order to get like a uniform kind of color representation, people perform stain normalization. One of the most popular techniques is Marchenko normalization, and a lot of papers that do weakly supervised whole slide image classification perform stain normalization as one of their pre-processing steps. The next step is to perform feature extraction, which basically you now have this stain normalized patch, and that is 224 by 224 pixels, usually three color channels, and you convert that image into a vector, for example, a 768 dimensional vector that just represents the content of that image. So you use a pre-trained feature extractor. Often these are trained with self-supervised learning on large data sets, and individually you extract for every tile this one vector that represents the tile. So for n tiles, you're going to get n feature vectors. And this is basically all the pre-processing. So this can all be done before you train your actual weakly supervised model. So during the training of the model, you can just load these vectors. You don't have to load all this other stuff, because that's what you can save to the disk beforehand. And here, there's also a big variety of options, or increasing variety of options for feature extractors. On the left, I'm showing some of the open feature extractors where the weights are released, and on the right, some of the larger foundation models, which are where the weights currently cannot be released or are not released. And in my study, I compare basically all of the openly available feature extractors. So for training, then, you take these feature vectors and you aggregate them. And the way that this is done is you have some sort of network that can take a variable number of features, and these n feature vectors are combined into a single feature vector, which could be of a different dimensionality. And finally, you classify that through a multi-layer perceptron or single linear layer, whatever, to perform the final output prediction. So that's the kind of more technical explanation of how you get from a whole slide image to the final prediction. As you saw in the three steps in the middle, so normalization, extraction, and aggregation, there's a bunch of different options that you can pick, and it's often difficult for a computational pathology researcher who just wants to train a whole slide image classification model to choose, to decide which of those choices is best. So for example, for the normalization step, you could perform stain normalization or you could not. You could even perform image augmentations, which, if you think about it, an image augmentation is or stain normalization is just one type of image augmentation. And then for the feature extraction, you've got the ImageNet pre-trained models. You can just take whatever, a ResNet 50 pre-trained on ImageNet, use that as your feature extractor, just cut off the final classification layer. Or you could take any of the pathology-specific models that were pre-trained on pathology slides, which are open, or you could use one of the foundation models if you have access to those. And finally, for the aggregation, there's also many options. A popular one is a tension mill. but you could also use transformer-based architectures or just simply average the feature vectors. And in my study, basically, we just compared all of these different options with the exception of the foundation models, which are not available. To go into more detail, these are the feature extractors that we compared. So we compared cTransPath, Ficon, which is from Oken, and then there's 3 models from Lunit. They performed a benchmarking study where they compared different models, different self-supervised learning architectures or methods for pre-training these feature extractors. So Dino, BT, and Suave, and then also we compared RedCCL. Basically, what these all have in common is that they were trained, so pre-trained on TCGA and possibly some other smaller data set in addition. But just for reference, TCGA has around 30,000 slides, whereas you have these larger foundation models. For example, the one presented just 2 weeks ago, which used 400,000 slides. So there is an order of magnitude or more difference between the models that I'm comparing and the largest ones that are trained but closed source or closed weights. All right, so our analysis looks at this from 2 perspectives. First, we look at the latent space of the feature extractors to gain an intuition on specific properties. And secondly, we perform like a comprehensive evaluation by training a bunch of downstream models on various downstream tasks in order to obtain empirical evidence in order to compare the feature extractors and see which aspects of this whole pipeline are most important for increasing the final prediction accuracy, or AUROCs. Starting with the latent space, we perform this visualization where we basically do a dimensionality reduction using t-SNE to visualize the latent space of, here I'm visualizing 2 different feature extractors. One of them is Lunit-Dino, which we found to be one of the best feature extractors. And on the right is the same deep neural network architecture, VITS, but using ImageNet weights and not the pathology pre-trained weights. And what we have here is every point corresponds to a patch in a data set. So right here, we're visualizing a colorectal data set that has 9 tissue classes, or actually 8. We removed the background class. And so every point represents to 1 feature vector, which corresponds to 1 patch, right? And then we draw a line from that patch, so from that point, to where that feature vector ends up if we were to perform stain normalization before extracting the feature vector. So think you have one original patch, you perform stain normal, as you perform feature extraction, that gets you a point in the latent space. So in this visualization. A bunch of long lines, and that means
Hi, George.
Yeah.
Oh, sorry, I think we lost you for about a moment, well, about a minute then. So, but we can hear you again now, so that's fine.
Oh, sorry. Sorry about that. I'll just start with this slide again. Okay, so yeah, so the latent space, we were visualizing the latent space, and the way that we do this is here I'm comparing Lunit-Dino on the left, which is one of the best feature extractors that we found, and VITS, which is the same deep neural network architecture, just pre-trained on ImageNet instead of the pathology weights, basically. And this visualization is a t-SNE dimensionality reduced version of the latent space. So every point in here is a point in the latent space. and we draw a line between the original point. So when we take a patch, we extract the feature vector from that patch, which gives us a point in latent space. And then we take the same patch, perform stain normalization, and then extract the feature vector. That gives us a second point in latent space. And we draw a line between these 2 points. So every line basically corresponds to a pair of an original feature vector and its stain normalized version. So having a lot of long lines means that performing stain normalization means that the features jump a lot in latent space, which would mean that the feature extractor is less robust to stain normalization. Yeah, when you look at this, actually, you would think that maybe you could think that Lunedino is not very robust to stain normalization, because you can see a bunch of longer lines than on the right for the ImageNet version. But actually, what we found to be more important is the fact that the jumps are not between the clusters. So right here, we're visualizing different tissue classes. So tumor, stroma. So every patch is classified as tumor, stroma, or whatever. And most of these jumps are within the clusters. So these lines are kind of staying within their clusters. But on the right, you see that they jump between clusters. So this is normal tissue might jump into the tumor tissue category. Yeah. Oh, by the way, please feel free to interrupt me if you have any questions. So yes. We can also do the same analysis with image augmentations. Here, I just show all the augmentations that we looked at. In fact, you can just regard stain normalization, so Marchenko normalization, as just yet another type of image augmentation. Yeah, so for that, I could plot 30 different latent space visualizations, but that would be quite difficult to analyze. So instead, what we did here is we looked at the distances of those jumps in latent space. So how much does the feature vector change when you perform some sort of augmentation? And here again, I'm showing those two feature extractors we just talked about, and I'm sorting the order of the augmentations by the Lunedino feature extractor. And you can see the smallest jumps are, or the smallest displacements in latent space are caused by augmentations such as lowering the contrast, flipping horizontally or vertically the patch, rotating it by 90-degree angles, and so forth. What you can see also is that the ImageNet feature extractor has larger the cosine distances, so the jumps are generally larger for the ImageNet feature extractor, which means that it is less robust to these augmentations. One thing we were quite surprised by is that rotating by random angles, so rotating the patches by random angles, actually causes large jumps in latent space. So we looked at that in a bit more detail. Because when you look at a pathology slide, there is no up or down, left or right. You can arbitrarily rotate a pathology slide, and the information is still the same. Unlike natural images, where if you flip an image of a beach, for example, upside down, that would make no sense at all. But if we have a pathology feature extractor, we would hope that the feature extractor would extract the same or similar features for a patch if it is rotated. So what we looked at here is, this is the visualization for rotating by 90-degree angle. And you can see for Lunedino, there's almost you can see no lines basically, because there are basically no jumps or very small jumps in latent space. However, for the ImageNet baseline, you can see some jumps. For the random rotation, you see this this visualization where there's a bunch of jumps everywhere, and we were at first quite confused by that. But then we had the hypothesis that this might be because when rotating by random angles, you obtain these black edges. And we actually found that that was the reason why we see these large jumps. So simply enlarging the image, so the patch, then rotating it, and then performing a center crop so that you're always sure that there's no black parts at the edges. You see that again for the strong feature extractor, there are no jumps in latent space, but then for the ImageNet baseline performs quite poorly. So this is not invariant to rotations. The second part of our study was to perform a comprehensive evaluation by training downstream models. To explain, oh, there's a question in the chat. Were the two feature extractors trained with the same data augmentation? The feature extractors themselves, we used pre-trained feature extractors. So are you talking about the training of the feature extractor? Because that probably used different augmentation. Sorry?
Can you hear me?
Yes.
Okay, great. Sorry. Yeah, I could ask my question live. Sorry. Yeah, the training itself of the feature extractor. So Dino, no, sorry, VIT and the other one, I forgot the name.
Yeah, so they use different augmentations because, well, first of all, we used ImageNet the VITS model we used was not pre-trained with self-supervised learning, just supervised on the ImageNet labels. So there's like 1,000 classes, and they've used different augmentations during training than Dino, of course.
Okay.
And actually, yeah.
Sorry, no, go for it.
Actually, one interesting thing which I will go into at the end is that this feature extractor performs pathology-specific augmentations in the SSL pre-training. So they really chose specific augmentations for like rotation to ensure that the feature extractor is more robust to, for example, rotation and other augmentations.
Because right now it's more like we're trying to find which feature extractor might be the best if you were to start from scratch now your analysis and choose one, but unless from the perspective of if you want to retrain your own, which type of data augmentation would be the best, that's why I asked that question.
Yeah, that is an important question, and there is some insight to be gained from that study on your particular question, which is using. So what we find is the feature extractors. So we looked at 10 feature extractors, and those that perform specific augmentations that are relevant to pathology tend to perform better in terms of the downstream performance. Not only in terms of looking at the latent space, but also the downstream performance. We find that, for example, including rotation in your augmentations helps the model performance downstream, which so you shouldn't just naively take however Dino or whatever other SSL method you used was configured for natural images and just apply to pathology, but it was better to use specific augmentations that are more relevant for pathology. For example, flipping an image upside down would not be a useful augmentation for natural images, but for pathology it would be useful.
Okay, thanks.
Yeah. Okay, so in order to quantify how these augmentations and stain normalization and the feature extractors themselves, all these moving parts, how do they affect downstream performance? We conducted a comprehensive evaluation where we looked at 5 different configurations of stain normalization or image augmentation. So We looked at performing no sort of normalization or augmentation. We looked at using patchwise or slide-wise stain normalization. We looked at using only rotation augmentations, and we looked at only using using randomly any of the augmentations. This is all during training of the downstream weakly supervised model. I should emphasize that the feature extractors and all that are frozen. Yeah, so we looked at 10 feature extractors, 3 downstream aggregation models that I mentioned earlier. We considered 9 tasks, which I list here. So we looked at 4 tasks in breast cancer, where we predict subtype and 3 mutations. And here we train on TCGA breast cancer and evaluate on an external dataset, CPTAC. We also looked at breast cancer lymph node status prediction, where we used the Chameleon 17 dataset, which contained samples from 5 centers. So we there we use 4 of those centers for training and 1 of them for testing to still ensure that it's basically an external set. And we do this 5 times to basically do a 5-fold cross-validation. And also for 4 targets in colorectal cancer, MSI status, and 3 mutations, again, training on TCGA and testing on CPTAC. And that yielded 6,750 trained models, which took around 200 GPU days if you were to train it sequentially. Sarah, yes, go ahead.
Another question, sorry.
Yeah.
Why not any outcome prediction?
I don't know how much information there is on outcome prediction in TCGA, but we did not do that. That would be an interesting step for future evaluation.
Yeah, I think you may have only overall survival, but it's still a start.
Yeah, that would be interesting to look at. Yeah, definitely. Awesome. So the results, what we found firstly is this is looking at stain normalization on the left, and what we plot here is the change in the test performance. We look at AUROC if you perform stain normalization versus not. So a positive value would mean that stain normalization improves performance, and a negative value means it decreases performance. And this is by feature extractor. And basically for all of the feature extractors, you can't see like a clear trend that stain normalization really improves performance. And especially for the 2 best performing feature extractors, so cTransPath and Lunedino, those the box plot shows even smaller variances and also no clear improvement. And the same actually goes for the 2 other configurations of augmentations, like rotating and flipping doesn't really improve, and also randomly applying all the augmentations. There's no clear trend, especially in the best performing feature extractors, that they improve downstream performance. This is averaged across all the tasks that we evaluated. So we performed like a bootstrap analysis.
George, may I ask a quick question here, sir, on the last
Yeah.
slide? So in terms of all augmentations, what sort of augmentations do we have there? And does that include stain augmentation? Sorry.
Yes, that includes sorry, no, that does. So stain augmentation, it does not include, it includes stain normalization as an augmentation, but does not include stain augmentation per se. And to be specific, these are the augmentations. Basically, for every patch during training at every epoch, you randomly select one of these augmentations to perform.
So in terms of using stain normalization as an augmentation, is that the case of you've got one target image and you're stain normalizing all the patches to that time?
To that one target image, which we chose before training. Yeah.
Thank you.
Okay, the relative performance comparison. So if another question we wanted to ask is, because we're comparing 10 feature extractors, which one is the best? And to do that, we compute this score, which we call the normalized differential Auroch score, which I will explain right now before we come back to this graph. So we were faced with the problem that we were analyzing or comparing 9 different tasks, and those tasks were of varying difficulty. So some were easy, for example, MSI prediction. So here are the raw results when you average across the 5 random seeds. And you can see, for example, MSI prediction is quite easy. You get Aurochs of 0.9 compared to, for example, predicting this mutation in breast cancer, where you only get around 0.6 Aurochs. So in order to be able to average across the tasks, we defined the score, which measures the relative efficacy of the feature extractors. And the way that we did it is basically we tried to answer the question with this score. If you were to train 10 downstream models, where each model uses the features from a different feature extractor, because we have 10 feature extractors, for a particular feature extractor X, how much worse is that test Auroch expected to be compared to the best out of those 10 models? This may be a bit confusing, so let me just illustrate this. Where are we? So let's consider an example. For a particular choice of augmentations, for example, if we always perform stain normalization, and a particular downstream aggregation model, for example, attention mill, and a particular downstream task, for example, predicting MSI in colorectal cancer, for this particular configuration, we have always trained 50 models, because there are 10 feature extractors across 5 random seeds, so we get 50 models. The way that we compare this is, if you take the 10 feature extractors and you randomly select 1 seed for every feature extractor, that would be 1 trained model. That each one of those models gives you a test Auroch. So this is just an example. So for example, for C trans path in blue, we select the first seed for that was the Swin transformer. For C trans path, we select the 3rd seed, and so on. So this is just a random configuration of seeds, and we get an Auroch score for each one of those. Then we compute the difference of the Auroch scores to the best performing model. So in our case, this model performed best with 0.92. So the difference to the best model is 0. And all the others have larger values because they are worse. So they have larger differences to the best model. And in total, you can arrange these different seeds in 5 to the power of 10 different configurations. And you then get 5 to the power of 10 different values for the differences for every feature extractor. And then you just average across. So you would get all the values for the first feature extractor, and you just average across all those. And that would kind of give you the score. This was all just a long, convoluted way to explain that just a lower score is better, and it is a fair way, in our opinion, to compare the feature extractors' relative performance, accounting for the variability in the seeds. Yeah, I think there's a question.
Yeah, so I didn't really understand. So this is Fayaz, and I've got a question on the evaluation. So you take different seeds and you evaluate each method with a different seed, and you get, let's say, 5 different AUs or seeds for a given problem, right? Did I understand that part correctly?
Yes.
And then how do you go from there to your normalized differential AUROC score? Is it taking the difference of each to the maximum value? Is that what you're doing?
Yeah, so in one particular configuration, you take the difference to the maximum value. But this is just one way of selecting a seed for every feature extractor.
Why should we select a seed? So seeds are there just to show that there should be variability across different runs, and we shouldn't be using it, or at least not selecting it as a sort of a pseudo hyperparameter of your experimental process. Or maybe I'm missing something.
Well, so okay, if you just take okay, firstly you could just take this table. This is when you average across the seeds, and now you have a bunch of numbers, and each of those has a variance, right? And how do you compare? How do you say across all these tasks, in general, which feature extractor is better?
So there
You
are tests
could.
for that, George. There's something called the Cochrane test, and there are a couple of other statistical tests that you can use to establish whether a result that you're getting, let's say if you are getting 0.9 plus minus 0.02 for one of the feature extractors, is that significantly better across all of the tests that you have done in comparison to another one. Fixing it just to the maximum runs the risk of biasing your results. If you get one fluke, or your method, like in the process, at one run, your method is by chance getting a very high value, then that is going to bias all of your analysis. So I'm not saying that's happening here. I'm just saying that that might be possible.
Yes, I think one thing to look at though is that because we're looking at all the different configurations, the 5 to the power of 10 configurations, and averaging across the configurations, you're basically eliminating all these outliers because you're averaging across all the different ways you can select a seed for every.
So I take it when you go to your ROC table, you've got the numbers there. If you could go back to that table, please, that you were just showing.
Yeah.
I think it's still taking some time. Yes, this one.
Oh.
So these are the results. So for example, when you use attention MIL with C-trans path features on the subtype prediction problem, you get an AU or a C of 0.82. Then if you use a transformer, you get 0.81. So if my question or hypothesis is transformers better than attention MIL, then what I would do is run a paired statistical test on the two to give me a p-value of what is the relative significance of this particular difference in results.
Yes. Yeah, okay, for here, I'm just trying to compare the different, if you select just one model, I'm trying to compare which feature extractor is best.
It's the same thing there. So if you want to test that hypothesis, then for attention MIL, you get the C-trans path. Let's say it gives you 0.82 plus minus 0.02, and then you get the vision transformer B, which gives you 0.74. Is 0.82 significantly better than 0.74? You can do a statistical test for that.
Yes, and you could probably do that for all the combinations or
well, you don't need to do it for all, because you have these average numbers. You can compare it across all of the different tasks, and you get a response. My worry is when you fix it to the largest value and normalize it to compute an ROC score, there may be a bias creeping in because you're just fixing at the largest number, which may be there just by chance. I'm sorry for taking this discussion. We can have a discussion afterwards as well. So if you want to continue, that's perfect.
Yes, I would be very interested. I mean, we discussed this internally as well a lot because we were trying to figure out the best way to evaluate the relative performances, and yeah, definitely. One thing I just want to emphasize is that we don't compare to the best Auroch in general, but just for every one of those 5 to the power of 10 configurations, for each one of those configurations, we compare to the best one inside that configuration. So it's not overall, but within the configuration. Yeah, okay, but I'll move on. So using this measure where lower is better, we found that C-trans path and Lunadino perform the best over when you average across all the tasks. So when you compare this normalized differential Auroch score and average that across all the tasks. and then we get these scores. So for every task, you then get a score. For example, in this case, in the case of attention bill for C-trans path for breast cancer subtype prediction, compared to all the other feature extractors, here actually C-trans path just in all of the different configurations always achieved the best performance. So here we can be pretty sure that C-trans path is the best. And we similarly do that for all the other feature extractors, and then you can just average that and get like an average performance across all the different tasks. And actually, we found interestingly, for example, that the results were quite similar no matter what downstream model you use. The best feature extractors are always C-trans path and LUNAT-DINO. And in general, C-trans path performed better on breast cancer, and LUNAT-DINO performed better on colorectal cancer. Just an interesting aside. Another thing we looked at was performing stain normalization on a slide-wise versus a patch-wise basis. Yeah, I think there's a question.
Yes, it's me again. Can you go back to the previous slide for a second?
Yeah.
There's a tiny lag for us, so I don't know if you.
Sorry.
Yeah. My question was, I mean, you mentioned something. You said you think that C-trans path is most of the time the best feature extractor, especially for breast cancer, might be less for colorectal cancer. I do not have the specificities on which those feature extractor were trained on. I mean, in terms of tissue type, was it the same data set, or it might be the fact that one was perhaps trained more on breast cancer tissue? And is there anything about this?
Well, so C-trans path was trained on the whole of entirety of TCGA, which I mean, the biggest cohort in TCGA is breast cancer. But it was also trained on additionally trained on an internal cohort from the authors, which I don't know exactly what the distribution of tissue classes, tissue types there is. So I don't know exactly about C-trans path. And then LUNAT-DINO was also trained entirety of just TCGA without, I think, without an additional internal proprietary cohort. But yeah, I think TCGA itself is biased towards breast cancer if you look at all of the slides. Yeah.
Maybe you should, I don't know, maybe you can correct from that to make sure that the improvement is due to the feature extractor itself and not the type, the tissue type the feature extractor was trained on. Because then the subsequent question is, should I just, okay, let's say I have the power, right? Should I just retrain on my own tissue, or eventually the weights that I can find online would be better? I think that's the most interesting question.
Interesting. Okay. Yeah. So looking at equalize kind of weighting the averaging of the tasks by the percentage of training data that belong to a particular tissue type. Yeah, that would be interesting.
Maybe check if there is a correlation between the numbers of seeing tissue types versus what perform better, and just to see if there's a sort of same trend, and then I don't know.
Interesting. Yeah, and I mean, one thing you could compare here is all the LUNAT models, because they were all trained on the same data set. Amongst them, there is no bias because they are all trained on the same data set, and the DINO model performs best.
Yeah, that's true.
Yeah.
Yeah. Okay, thanks.
Okay, are there any other questions?
Just a very quick observation, if I ask again. So
Yeah.
if you look at this, for example, C-trans path at the bottom under subtype gives you, like, it's the highest one, so you get 0.00 plus minus 0.00 for that, right? Now, let's CCL for this one gives you 0.01 plus minus 0.00. So roughly, they are both the same. And I believe if you conduct a statistical test on them, they're like this. One may come out statistically significantly different from the other, but practically they aren't that different. That's my point about using the max. So if you just focus on the max, we might be biasing the results here. And there are statistical tests that you can do to compare. It's not wrong. I'm not saying this is in any way, but it just would be statistically more robust if you do a pairwise comparison across different tasks that you have here.
Yeah, and I mean, you could in the end probably also do it across the averages here. I don't make any, so in the paper we don't make any claims about the statistical significance of the differences, but that could be a thing to look at in future. Okay. Another thing we looked at is whether or not you should performing slide-wise stain normalization is better than patch-wise. So you could calculate the stain matrix when you perform stain normalization on the entire slide or just on a per patch basis. When you perform it on the entire slide, that is computationally a lot more expensive. But we found that there was no real benefit of doing either. So both of them don't really improve the performance significantly or decrease the performance significantly. So it's in both ways. And finally, we compared the three different downstream aggregation models by just using the same normalized differential Auroch score, but pivoting it. So now we don't compare the feature extractors, but we compare the downstream aggregation models, and we don't find a clear trend which one is a lot better. You can see that the error bars are quite high. But in general, we find that there's maybe a slight signal that attention mill is the best option. Finally, I'll just quickly show so all the results. So mainly the results I showed those graphs earlier were looking only at the attention mill as a downstream aggregation model. But if you also look at the transformer or mean pooling, you get similar results in terms of the relative performance comparison, in that cTransPath and Lunedino perform best compared to the other feature extractors, and that there is no observable benefit in performing these stain normalization or augmentations. One interesting thing is that when you perform mean pooling, you get a lot less variance in the observations. Okay, to conclude, the main things we found is that the choice of feature extractor is the most significant factor for improving downstream performance. So the biggest leverage you have to improve the performance is choosing a good feature extractor. And we found that cTransPath and Lunedino tend to perform best. Both of these actually perform pathology-specific SSL techniques. So they they perform specific augmentations that are relevant to pathology, for example, rotation and various stain augmentations. So probably in training your own SSL feature extractors, or in future, it would be beneficial to look at what they did and see what other ways we can improve the pathology incorporate the pathology domain knowledge in the training of the SSL feature extractors. Secondly, we found that the choice of downstream aggregation model is not as important for downstream performance. There was a weaker signal there. And finally, we find that stain normalization and image augmentations don't significantly affect the performance of the downstream models. Some ideas for future work, or current work actually, is to compare more feature extractors. I've just finished running some more experiments with additional feature extractors. There was another model released by Oken. So Oken released two versions of their model, the FICON model, the student and a teacher model. So we only looked at the student model, but now also perform more experiments with the teacher model. However, we found no significant benefits of the teacher model. We also want to compare across different magnifications. So all of the data I presented today was with an MP microns per pixel of 1.14, which was what was default at the lab at Kata lab, which corresponds to around about 8 times magnification. But now I reran most of the experiments again with 20 times magnification. So I'm going to report on that soon, what we find there, but the initial results are still, we find similar results in terms of the quality of the feature extractors, as in, at least for breast cancer, we still find that C-transpath and Lunatino perform best. I haven't finished the colorectal cancer experiments yet. Also, it would be interesting to look at more downstream tasks and different cancer types. Right now, we only looked at 2 cancer types. But this is all, of course, requires a lot of different, a lot of computational resources to run these experiments. And finally, the one question I'm thinking about, and might be also an interesting discussion, is to find out some way of quantifying the quality of a feature extractor without needing to train a bunch of models. If we could just somehow analyze the latent space, find some sort of metric in the latent space that might provide some indication on the quality of the feature extractor without needing to train the downstream models. So with that, I would like to thank you for your attention. We've got a project page where we're releasing all the data and all the raw, so the raw data and code, so all the individual predictions. So the work is reproducible, and you can actually probably perform any sort of statistical tests afterwards as well to see if you're not satisfied with the statistical or the way that we analyze the results. Yeah, I think there's a few questions which I would like to take.
Fantastic. Well, thank you very much, George, for a really interesting talk. It was so. Yeah, with that, we'll start with some questions then. Does anyone in the room have any questions before we ask people online? Fab. So, oh, we've got a question from Mark.
Quick one. I was just going to say, given kind of the added complexity that doing state normalization adds to pipelines, in the light of your results, which say it makes very little difference, would you say from your results you can suggest that perhaps leaving that out of your machine learning pipeline is actually a good idea?
Yeah, I mean, that's what I was trying to say. Actually, I didn't talk about the computational overhead, but yes, it's significant. So I think it would be feasible to leave it out, or at least whenever starting a project, just not do state normalization. Maybe afterwards, see if you have the computational resources. Once you've got a minimum viable product, basically just see if adding state normalization improves the results. But I think to just get something up and running, I think we're fair in not just not performing state normalization.
Thanks, George. A bit of a follow-up question from me for that then is, so we've seen a few more recent papers also suggesting that state normalization isn't necessarily a good first step, but that potentially things like state augmentation might be better as a processing step. So would there be any plans in the future to look at trying to see the effect of state augmentation on your different, on the choice of your feature extractor?
That might be an interesting thing that I haven't planned that yet, but I think that would be an interesting direction to look at. So looking at state augmentations as well as a kind of pre-processed augmentation. Yeah, definitely.
Brilliant. So we've got another question from Sarah online who's asked. Of course, you can, Kelly.
Yes.
I'll just yeah, go on you first.
Hi, George. It's a really good presentation. And I would like to ask, you have mentioned in the data augmentation part, you doing different type of augmentation for the image patches. I was just curious, if you rotate the patch like to a certain degrees and you pad the patch with the 0 values, right? And you claim that the feature extractor results is got decreased. Would you mind to explain a little bit on that? Why were we padding with the 0 value to the rotated image patch and the results is decreased a little bit?
So are you asking why this decreases the accuracy of the feature extractors, the black edges?
Yeah.
But I can't answer that, unfortunately. I don't know. I think it's probably because the SSL feature extractors never saw these sorts of black edges during their training, so they don't know how to handle that. Because usually, I mean, you of course have these augmentations where you rotate the image, but then afterwards you always perform usually some sort of center crop or anything so that there is no black areas. At least that's how I understand.
So
how I
it's without padding with 0, you just provide the whole image instead, right? Well, I mean, when you rotate the image, do not padding with 0 value, or the black value. Is that what you suggest in your research?
Yeah, well, I'm suggesting that. Well, first of all, I'm suggesting to not perform image augmentations. So don't augment your images, because that actually increases the computational complexity even more. Because for each augmentation, you're going to have to run that through the feature extractor as an additional pre-processing step, where usually you only need to run your original patch and get the embedding vector. If you do image augmentation, you then for every patch maybe get 30 different feature vectors because you're doing 30 different augmentations, and you need to do that all before training. So that's significant computational overhead. And then I would just suggest not to perform the augment. If you were to perform augmentations, don't do the random rotation where you then get the black edges by padding it with 0 pixels. You could, if you wanted, just do this random rotation, but then perform a center crop so that you don't obtain the 0 values.
Got it. Thank you.
Yeah.
Brilliant. Thank you. We've also got a couple more comments online. So Sarah's asked, said, very nice project and results. Just being curious, how long did it take to run all of this? I think you said it was about 200 GPU days, was it?
Yeah, so it was 200 GPU days to train the downstream models, but that does not include the feature extraction and state normalization, all the pre-processing itself. I didn't actually calculate how long that took, but that was also a significant amount of time. Yeah, the project took several months. Of course, the 200 GPU days wasn't run all on one GPU. It was parallelized across multiple GPUs, so it was faster. But yeah, it took quite a few months to run all the experiments.
Fantastic. We've also got a few more comments online. So Faisal said a good contribution in terms of the breadth of analysis. Nice work. And then he's got a few other suggestions. And John's also asked, what dimensionality reduction technique was used on feature extraction latent space, and does it attempt to preserve latent space distances?
Yeah, that's the excellent question. We used t-SNE for the dimensionality reduction, and we used the cosine distances as the distance measure, so not Euclidean distances, because we used cosine distances everywhere in the paper for the analysis. But in the initial stages, we actually quite extensively compared using cosine distance or Euclidean distance, even Manhattan distance and other distances, and found that they all basically showed the same thing. So the results look quite similar. And also we used UMAP as well and found also no large differences in what the results showed. But for the paper, we just stuck to t-SNE.
Brilliant, thank you. I think we've got time probably for one final question. So I don't know if anyone online or anyone in person's got a final question. Alternatively, I'll
I was going to ask, what difference does patch size have on the different feature extractors?
Did you hear that question? Sorry, George.
Yes, what's the difference? What effect does patch size have on the performance, basically? We only looked at a patch size of 224 by 224 pixels, and because that was also what all the feature extractors that we compared used as the input dimension. So I can't speak to that.
Brilliant. Well, thank you very much then. I think we'll draw the seminar to a close with that final question. So thanks again, George, for joining us. It's been a really interesting discussion and presentation. And for a reminder for everyone else online, so our next seminar is on Monday, the 19th of February at the same time from 2 till 3. I'm delighted to be joined by our very own Dr. Mark Eastwood, who will be presenting updates from the TI Toolbox on TI Viz. So I hope to see everyone there. But thanks again, George, and thank you for everyone in person and online. And I hope everyone has a good.
Thank you.
Thanks, George.